Hello, welcome to the instruction video on enzymatic activity of manganese peroxidases in soil. Manganese peroxidases are extracellular enzymes belonging to the group of oxidoreductases. Along with lignin peroxidases, manganese peroxidases are categorized under lignolytic enzymes and can aid in degradation of many hazardous organic compounds. Manganese and lignin peroxidases are heme proteins consisting of protoporphyrin 9 as a prosthetic group. Manganese peroxidases are glycoproteins and its structure has two domains, with the heme ionic group in the middle, 10 major helixes, a minor helix, and 5 disulfide linkages. Manganese peroxidases possess specific linkage stakes in the manganese bonding site, which distinguishes them from other peroxidases. Manganese peroxidases are besides other involved in the degradation of polycyclic aromatic hydrocarbons. And how to measure the activity of manganese peroxidases in soil? First of all, you need to prepare chemicals which will be used during extraction and measurement. There are solutions of phosphate buffers. The first 50 millimole phosphate buffer will be used for the extraction, and the second 100 millimole phosphate buffer will serve to dissolve the DMAB. The reaction of MbTH and DMAB in the presence of hydrogen peroxide, manganese cation, and manganese peroxidase gives a deep purple-blue color, so the DMAB and MbTH are the coloring agents for the enzymatic reactions, while manganese sulfate, hydrogen peroxide, EDTA, and water will serve to distinguish among manganese peroxidases, and oxidases during analysis. As the manganese peroxidases are extracellular enzymes, soil samples can be analyzed in a freeze-dried form. Soil should be ground and sieved through a 2 mm sieve prior to analysis. The common mass of sample for the analysis is 200 mg. Plastic Erlenmeyer flasks are suitable for the extraction. When the sample is weighed, 20 ml of the 50 millimole phosphate buffer is added, and the mixture is briefly homogenized using the high-speed batch homogenizer. The homogenizer must be washed using demineralized water after each sample to avoid cross-contamination. Right after homogenization, sample suspensions must be filtered through filter paper in order to remove solid particles and obtain a solution where enzymes are dissolved. The step of desalination follows the filtration. To do so, desalting columns are used. The principle of desalting is based on size exclusion chromatography, so the molecules are separated on the basis of differences in size. You can see the working procedure on the screen. Pipette is set to the volume of 3.5 milliliters, and demineralized water is introduced to the columns. This water serves to flush the columns. When water stops dripping, the sample can be introduced. .5 milliliters of the sample solution is pipetted into the column. The leachate is still not collected because flushing water is pushed out from the column by the sample solution. When columns stop dripping, test tubes for sample collection are placed under the columns and 3.5 milliliters of water is added to the columns to push out the desalinated sample solution into the test tubes. Finally, 3.5 milliliters of water is pipetted to the columns in order to wash the columns. Each sample in each microplate will consist of 3 sections where different enzymes will be measured. To do so, 3 solutions of distinct composition must be prepared. Solution AR will be used for determination of manganese peroxidases, solution AP for total peroxidases, and solution AB for oxidases. The composition of each solution is shown on the screen. Volumes shown are calculated for 1 microplate, where 8 samples can be analyzed. The mixing of pre-prepared solutions is done in the labeled reagent reservoirs by pipetting the given volume of each solution. Here you can see the design of microplate, where 8 samples are analyzed. It is important to measure each enzyme in at least 4 replicates. Each sample is represented by 1 column. Start with the transferring of the sample extracts into clean and marked region reservoirs and pipette into the microplate according to the scheme. Change the pipette tips between the samples. Next step is to distribute the substrate solutions into the microplate. 200 microliters is pipetted into given wells. Right after pipetting the last solution, microplate must be placed into the reader to start the measurement. The principle of the measurement is based on color change of the solution caused by the reaction of DMAB and MBTH. The reaction is catalyzed by the enzymes of interest. The resulting color is purple-blue, so the absorbance is determined at 590 nanometer wavelength. The measurement is done in 7 cycles after 2 minutes each. It takes around 12 minutes to analyze the whole microplate. Now is the time to answer displayed questions. In case you miss an answer, we recommend watching the video again.